Okay, good morning. We're going to go ahead and get started here. Does anybody have any questions? Let me say the same thing. Does anybody have any questions on anything? Okay, we're going to continue on in our discussion on the cell and see how far we can get today. If you have any questions on exams, lab exams, lecture exams, lab content, shoot me an email or ask me a question in the middle of class. If you have any questions on the content we're going over, ask me a question. I'd be glad to answer it. So, and if you do send me an email, I will get back to you right away so that you can get an answer. If you're having trouble with your lockdown browser, let me know. Because I know a number of people have had problems with their lockdown browser. I don't know why. I don't know the workings of the browser, but I know that we've had some issues with it. So just let me know if you're having a problem trying to access your test. Is the bones test still due October 5th, or will it be pushed back to? I'm probably, I'm thinking along the lines of pushing it back. I'm thinking that way. I haven't said yes or no, but I'm thinking along those lines. So you'll know. I'll make a decision today on whether or not we should push it back. So, but it's still only September, but we're ahead of schedule. So I think if we wanted to, we could push it back. So anyway, I'll let you know. So that's the best answer I can give you right now. Let's see. Okay, so let's go ahead and get into cells. Lab this afternoon, by the way, will be the appendicular skeleton. So go on from here. Now let's bring this up. Everybody should see the screen that says tonicity. And tonicity is not something that anybody runs up to you and says, "Hey, what about tonicity? Isn't that exciting?" Well, we don't think about it, but it is pretty important to us. Tonicity has to do with the concentration of the solutes outside the cell and inside the cell. Now, the principal solute we pay attention to is sodium. Sodium levels, the amount of sodium in our bodies usually doesn't change all that much. Water levels vary all the time. And what our body does is it monitors sodium levels to tell us indirectly what our water levels are. If our salt concentration goes up a little bit, it's usually because we've lost a little water. If we get thirsty, we're thirsty because we detect a rise in salt. Now, we aren't retaining salt, we aren't adding more salt to our bodies. The only way salt levels go up is if water goes down. Consider a beaker of water. You have a beaker of water, it contains a liter of water. 1,000 milliliters of water is a liter. And you add 10% salt to that. Make it 100 grams of salt and make it a 10% salt solution. So great, now you've got a beaker full of salt water, 10% salt. If half the water evaporates, if the water evaporates down to the 500 milliliter mark, now you put 100 grams of salt in there to make a 10% salt solution. The water evaporates down to the 500 milliliter mark. You lost half the water, but you haven't lost any of the salt. You don't lose salt through evaporation. So you still have 100 grams of salt in that beaker, but now you only have 500 mils of water. Now you have a 20% salt solution because you lost half the water. And eventually that water will get saltier and saltier as more and more water evaporates, so you have nothing left in the beaker except your 100 grams of salt. And so our bodies are like that. We generally don't see a rise or a drop in the amount of salt we have present, but the concentration of the salt in solution is based on our water levels. If we drink a lot of water real fast, well, if you finish a marathon, you chug down 6 liters of water very quickly. Not a good idea, but you do it. Where does that water go? It goes into your stomach. It goes into your small intestine. From there, it goes into your blood. Everything we eat and drink goes into your blood. And so now you dilute the plasma. You add all that water, goes through the liver's fine with it, goes into your plasma. You have diluted your plasma. The salt concentration of your plasma has dropped because there's an excess of water. What happens to that plasma? It leaks out into the interstitial spaces around your cell. The salt concentration in the interstitial fluid outside of the cells is lower than inside the cells. Water will, if you drink 6 liters of water, that water will ultimately go into the cells and cause them to swell up because you didn't maintain the same concentration. Because that water that you drank, that 6 liters of water that you drank, goes into your blood. It leaks out of your blood into the interstitial spaces and dilutes the water that's already there around the cells. Salt concentration is lowered simply because you diluted it. If you take back to my beaker example, you take a beaker that contains a liter of water, 1,000 milliliters of water, and make it 10% salt by adding 100 grams of salt. 100 is 10% of 1,000. So you have a 10% salt solution. What if that's a big beaker and you can add another liter of water to it? You've diluted that salt concentration where it's not 10% anymore, it's 5% because you've doubled the water. When you increase the water concentration outside of our cells, you lower the salt levels. Salt is a solute, and when you lower the solute concentration, water will leave one side of the membrane with the highest water and go across a membrane to the side with the lower water. So it's important that we don't want water to move back and forth like that. That's not a good thing for cells to expand rapidly or to shrink. So we try to maintain the concentration inside the cell and outside the cell at all times. And that's an isotonic environment. That way we don't have any movement of water. We don't like to see water moving back and forth. We don't like to see cells placed into a high salt solution. When a person becomes dehydrated, the water we're losing is from the plasma and the interstitial fluid. So the salt content, when a person becomes dehydrated, they've lost water. The salt concentration outside the cells goes up because the salt doesn't disappear when we're dehydrated. The salt stays there. We just lost the water. It's like the water evaporated. It's like the water evaporated. So now the interstitial fluid is saltier than the cell. When a person's dehydrated, the cells give up water to try and reestablish equilibrium. The cells will shrink. That's what's called a hypertonic solution. And if we drink a lot of water, again, we create what's known as a hypotonic solution, where the salt concentration is less than inside the cell, and water goes into the cells, and the cells can swell up, and some of them can pop. They can lyse. If they shrivel, it's called crenation. If they pop, it's called lysing. They rupture. They can't recover from rupturing. They can recover from crenation, but they can't recover from rupturing. In our bodies, we like to maintain equilibrium with our with the concentrations, with our salt concentrations. If it's 0.9% inside the cell, we'd like it to be 0.9% sodium chloride outside the cell. We use isotonic solutions, bags of IV bags, intravenous bags, normal saline, the same salt concentration and makeup as what's in our plasma. Hang a unit on somebody like that, goes into the vein, and it is a fluid expander. It increases the fluid volume of the plasma. And it's also, we use it as a line to get medications in. Now, occasionally, if you have a patient that is swollen because of water retention, they're retaining lots of water. Perhaps they have congestive heart failure. Perhaps they are having an allergic reaction to peanut butter or to a bee sting or to seafood, a shellfish or something. Well, we don't like any of that. One of the things that happens in those conditions is our cells retain water. So how do we get the water out of the cells? If you administer a hypertonic solution that is saltier than what's inside the cells, the water will be drawn out of the cells into the interstitial spaces, and we reestablish equilibrium. The reason we have to do that is because that water that's in the cells came from the plasma. So we suck water out of the plasma and send it into the cells. We reduce our blood volume. We don't like that. And something that you never do is give your patient an IV of distilled water, because that creates a hypotonic environment. And that water will enter the cells, and the cells will rupture, particularly red cells and the white cells. Okay. So enough about that. We're still talking about movement across the membrane here. I know we used to do this in lab when we were on the ground. We would take blood samples. I would lance myself, and I'd put a drop of blood on the microscope slide, and we didn't get anybody. We couldn't take a volunteer, but I put a drop of blood on the slide, and I'd add some salt water to it, and we'd put it on the camera, and you could see the cells shrinking, crenation, and they would, if you put normal saline back on them, they would recover the shape. If you put a drop of blood on the slide and added distilled water to it, 1 second you can see the red cells floating in the plasma. The next second you see nothing except a red smear because the cells all ruptured. Cells don't recover from lysis. They don't recover from being broken apart. Anyway, all of these approaches that we've talked about here are passive. They're free. Movement of, osmosis is a type of diffusion. Movement of water from high water to low water. Simple diffusion from high concentration to low concentration. Facilitated diffusion, we use a channel or a carrier to get substances across the membrane from high to low. What happens though when we can't get things across the membrane we need? What happens when we have a substance, a solute, that's too big for a channel or a carrier protein? Perhaps what happens if it's not soluble in lipids? It's water soluble, and we can't, but it won't go across the membrane. And what happens if there is no concentration gradient for it to go down? What happens if the concentration of a substance inside the cell is higher than the outside, and we still want to bring more substances in? We have to, what if we have to go against the concentration gradient? What if we have to essentially pump uphill? We have to work harder. And to do this, to do any of these three conditions requires energy. Energy in the form of ATP, because ATP is our medium, our currency, our money that we use in the cell to do things, to do work. We take it from glucose, we release the energy stored in glucose, we break the covalent bonds of the valence electrons. We take as much potential energy as we can use, and whatever of that potential energy that we can keep, we get to use briefly in the form of ATP. Say briefly because each ATP molecule only lasts about 7 seconds. Then we have to make it again, make it again, and make it again. We have active transport across a membrane, usually against its concentration gradient. We have vesicular transport, where we're moving stuff in bubbles that we call vesicles, either into the cell or out of the cell. So looking at active transport actually requires the use of a pump embedded in the cell membrane. The pump uses ATP to pump substances either one way or opposite directions. An antiport will pump one substance one direction and another substance the other way. So it's like a two-lane highway where traffic's going in two different directions. So that's what an antiport does. A symport brings transports two different substances together across the membrane in the same direction. But they are going against their concentration gradient. They're going uphill, if you will. Because when you're using active transport, you are pumping from low concentration to high concentration. You're going uphill. It's not the free ride going downhill from high to low like you get with diffusion and osmosis. Here you have to go from low concentration to high. If you have a lot of sodium outside the cell and a little sodium on the inside of the cell, but the cell doesn't want any sodium on the inside, it has to pump it out. It's not going to diffuse out on its own because you already have, most cells in our bodies have a high concentration of sodium on the outside. Sodium will continually leak into the cells, but it will not leak back out because the concentration gradient for sodium in the cell membrane is from the outside to the in. But you want it out, so you have to pump it out in here. And this takes energy in the form of ATP. Now, active transport comes in two versions. Primary active transport, where you're physically pumping across the membrane using ATP. Secondary active transport is a free ride. With secondary active transport, you take advantage of the concentration that you're maintaining outside the cell. I'll get back to that in a second. So primary active transport, you have a protein embedded in the membrane. It's a pump, maybe an enzyme, it's a pump, and it will change its shape. And you can remove substances from the inside of the cell membrane and kick them to the outside, going against the concentration gradient. You can take substances from the outside and bring them into the cell, going against their concentration gradient. We change the shape of the pump to accommodate the movement of these substances. We have different types of pumps. We have the calcium pump. We find calcium pumps in our muscle cells. We find calcium pumps in our cardiac muscle cells. Proton pumps. We find proton pumps in our stomach. Proton pumps pump hydrogens outside the cell. This is, hydrogen ions are what makes things acidic. The more hydrogen ions you have, the more acidic you are. Proton pumps are busy making hydrogen ions because we need hydrochloric acid in our stomach to help digest protein. These proton pumps, if they overproduce, it's the primary cause of acid reflux. So, we use proton pump inhibitors to control them. If you know anybody that takes Protonix or Prevacid, or maybe even some of the other ones, that if it says a proton pump inhibitor on the box or on the label, that's what it's doing. It's shutting down these pumps embedded in the membranes of the cells. And the most well-known of all of these active transport pumps is the sodium-potassium pump. Embedded in the cell membrane, it's an enzyme. It is an antiport because it pumps sodium out and potassium in. Let me give you a little bit of background here. All of our cells in the body have charges on them. We all have a measurable charge. Muscle cells have a charge of minus 90 millivolts. Nerve cells have a charge of about minus 70 millivolts. It's caused by the difference in concentration of sodium and potassium ions. Most of the cells in the body have very high concentrations of sodium outside. That high concentration of sodium outside is giving the outside of the cell its positive charge. Embedded inside the cell are high levels of potassium. These are the two key ions that deal with the charges on our cells, sodium and potassium. Well, the problem with sodium is it's such a high concentration outside the cell, it's always leaking in. There are sodium channels that and carrier proteins for sodium that allow it to leak in across the membrane. Potassium is always leaking out. It has its own channel proteins or carrier proteins that will allow it to leak out of the cell. If the sodium concentrations inside the cell reached equilibrium with the outside, if they became balanced, if the potassium levels became balanced inside to outside, the cell would cease to function. We can't let that, the cells can't let that happen. As long as the cell's alive, we got to keep high sodium high sodium outside and high potassium inside. So when sodium leaks in, we pump it out. When potassium leaks out, we pump it back in, and we use the sodium potassium pump to do that. Because when the sodium potassium pump pumps sodium out, it's then in a position to take potassium back in. And here's what the pump looks like down here at the bottom, embedded in the membrane. It will, when it opens up to the inside of the cell, it has receptor sites for 3 sodiums that will attach themselves there. Now the pump will change its shape and pump the sodiums out thanks to the presence of ATP. The ATP the pump will close the inside opening. It will close this side and open to the outside. Excuse me, and pump 3 sodiums out. Now that it's left open, it will grab onto 2 potassiums and bring them back in. And now it's got to reopen, ready to do it again. So it opens to the inside, picks up 3 sodiums, is charged up by the ATP, pumps the sodiums across the membrane, opens up its outer door. Potassium finds its way in here and lands on special sites just for the potassium. And then when they land on their sites, it closes the outer door and opens the inner door and releases the potassium to the inside. We need this for our muscle contractions. We need this for our nerve impulses. Sodium and potassium is critical. So we have to keep sodiums outside and potassium inside. And since they are an antiport, they're going in opposite directions. So if the greens are potassiums and the yellows are sodiums, notice how the pump, when it opens to the inside, will pick up 3 sodiums and an ATP. That allows it to open to the outside, close the inner door, open the outer door, the sodiums are kicked out. And while it's open to the outside, here comes some potassiums. That causes the inner door to open and the outer door to close. So we keep pushing sodiums and potassiums in and out of the cell. This is an important thing to remember. We're always pumping more sodiums out than we are bringing potassiums back in. 3 sodiums go out. Remember that. 3 sodiums go out and 2 potassiums come in. I guarantee you'll see it on a test question. You'll probably even see it on the final exam. It's one of those little factoids that you'll probably never forget, but you'll never need it. But we do need to know that sodium potassium pumps work. We got to keep the sodiums on the outside. Now, one of the benefits of pumping the sodiums to the outside is that we keep a high concentration of sodium out here in the interstitial fluid. That's what we want. We need it. First of all, we need it to keep the charge in the membrane maintained at minus 90 millivolts. Got to have that. The other thing it does is it sets up a situation of secondary active transport. So, here is our primary active transport right here. There's the sodium potassium pump. Three sodiums go in here and they go to the outside, and the pump is open to the outside. And there are receptive sites for potassium here. They land in here, potassium comes in. We keep the concentrations of sodium high out here because there are also symporters, pumps that, there are passive carrier proteins that will allow two different molecules to come across the membrane at the same time. One can't go across without the other. The presence of both of these molecules in this symport causes it to open up to the inside, release the contents, and then causes it to close the inside door and open the outer door. And this is called the sodium glucose symport. When sodium and glucose are present, when sodium and glucose are present here, they can use this symport. Sodium lands on the symport, opens up the presence of sodium on the symport, opens up the receptor site for glucose, and then the whole symport opens up and sodium and glucose go into the cell. Cell wants glucose. Cell doesn't particularly want sodium. The sodium allows the glucose to piggyback with it going in. Then we have to turn around and pump the sodium back out. By continually pumping the sodiums out, we provide a means for glucose to enter the cell. Now, is this how insulin works? Is this the carrier protein that insulin uses? I don't know. It may be. So, but this is a way we get glucose into the cell. Because remember, glucose is a big molecule. It's too big to diffuse through the membrane. It's also water soluble. It has to have some sort of channel protein or carrier protein to get it across. Is this the mechanism we use? Is it dependent on the presence of sodium? And if it is, it's important then that we keep pumping sodium to the outside. It's so important for a cell that a typical cell will spend 40% of its energy, its entire energy budget. It spends 40% of its energy budget pumping sodium and potassium back and forth. So that's a lot of energy to maintain this concentration, to maintain the charge on the cell membrane. Okay, so now that you've learned that, there will be occasions where it may come in useful about sodium and potassium, because if a cell's dying, it's not going to doesn't have the energy to pump sodiums out. And once we reach equilibrium with the sodium, then the cell's not going to function anymore. Okay, other ways to get across the membrane that take energy: vesicular transport or vesicle transport. A vesicle is like a bubble, but it's a bubble made of the phospholipid bilayer, just like the cell membrane is. We can bring stuff into the cell through what's known as endocytosis, endo for entrance. We can get stuff out of the cell through exocytosis. In both cases, it involves, in endocytosis, our cell membrane reaches up and grabs a hold of a bacteria and engulfs it in its own bubble. The bubble is made from the cell membrane. We bring it into the cell. It's contained in its own vesicle made of the phospholipid layer, and then that vesicle fuses with a digestive enzyme vesicle, and it's destroyed or eaten or whatever. Think of an amoeba going up and surrounding something, like a piece of food or a bacteria or whatever. In this animation, in this video up here, you can see a white cell, a white blood cell comes up and engulfs a bacteria, surrounds the bacteria and completely destroys it. It takes it into the inside the cell. It doesn't let the bacteria run freely through the cytoplasm. It keeps the bacteria in a vesicle, a sac, a bubble, and then it destroys it with its own enzymes. 3 types of endocytosis: phagocytosis, cell eating, pinocytosis, cell drinking, and receptor-mediated endocytosis, where we're only going to bring in certain things. Exocytosis is a lot easier. A vesicle that is already formed inside the cytoplasm of the cell fuses with the cell membrane, like we're seeing here in this bottom picture, and the contents of the vesicle are released to the outside. This is how we release proteins, for example, when they get made. So examples here: phagocytosis, the cell membrane reaches up and engulfs the food. Maybe it's a worn out cell. Maybe it is a cell fragment. Maybe it's a organelle that's breaking apart. When cells die, all their friends and neighbors come over and eat them. So it's very tough homeowners association here. But we engulf them, just like we saw the amoeba do. We engulf the particle and bring it inside in its own little vesicle, or vacuole, they may call it. It's surrounded by a phospholipid membrane, and then it's going to get dissolved by lysosomes. Same with fluids. We can pinch off around the droplet if we want. The same with receptor-mediated endocytosis. It's specialized. We bring in the specialized substance that we want. Exocytosis is different. It's much simpler. We form something inside the cell and we release it to the outside. The vesicle fuses, the vesicle containing whatever we've made, let's say it's protein, fuses with the cell membrane and then opens up on one side. So the contents of the vesicle are exposed to the outside of the cell, but the cell membrane is never compromised. The integrity is maintained, so the cytoplasm is never exposed to the outside of the cell. Here we see in this picture on the right, this is a vesicle releasing something, maybe proteins, outside the cell. The membrane here has stayed intact all the way around. Membrane has stayed intact, but we've opened up the vesicle to the outside. So all of these require the use of ATP. So now within, okay, so that was the membrane. Within the cell, we have the cytoplasm. The cytoplasm is made up of the cell sap, called the cytosol. Some texts are changing the name of cytoplasm to cytosol completely. The cytoplasm or cytosol has all the organelles contained there. It has the consistency, sometimes, we call it the cell goop, the cell sap. It may have the consistency of hotcake syrup, if you will, maybe, sometimes it's like a gel, but all the organelles are held in place there, along with proteins and sugars and salts and, droplets of fat and stuff like that. The organelles are structures. They're like little organs that do things for the cell. Many of them have the same phospholipid bilayer as the cell itself does, and they're all very specialized to do things. We have the mitochondria, the Endoplasmic Reticulum, the Golgi Apparatus, Peroxisomes, Lysosomes, Ribosomes, cytoskeleton, and centrioles. So what's the mitochondria? Well, a lot of textbooks like to call it the power plant of the cell. And I really don't like that term because it implies that they're busy generating electricity or energy. They're not generating energy. They are oxidizing glucose. They are burning glucose to release the energy contained in the covalent bonds of the glucose molecule. That's what mitochondria does. And it produces ATP. That energy isn't being generated. You know, like it's not being, we're not making energy. We are releasing it, converting it from its potential energy in the glucose molecule to the kinetic energy or the potential energy stored in the ATP. It's just semantics, you know. Anyway, this is where it happens. Mitochondria are where we oxidize most of our glucose to release most of the energy available to make most of our ATP. Because we make a little ATP without using oxygen. Not much, just a little bit. But the bulk of our ATP production occurs in our mitochondria. So it is the energy conversion site of the cell. And it's a double layered membrane. And it has, not only is it double layered, but it also has an inner membrane and an outer membrane. It's the inner membrane where all the work takes place on here. That's the mitochondria. The ribosomes don't have any membrane. They are made of RNA and protein. But ribosomes are where we make, where proteins get assembled. Ribosomes are where we find amino acids being brought together in the specific order dictated by the DNA. At a ribosome, we bring there are only 20 amino acids. So at the ribosome, we will bring amino acids together to form proteins in the order that is specified by our DNA molecule. So whatever amino acid we have, there's 20 amino acids, whatever order that we need to have to make our protein, is going to be brought together. The amino acids we brought together in that order at the ribosomes, and that's where we make our proteins. And they are usually attached to a series of tubules known as the endoplasmic reticulum. The rough endoplasmic reticulum and the smooth endoplasmic reticulum are tubules, tubes, tunnels, whatever, that completely surround the nucleus. Not like it just shows here, we're only seeing one portion of it, but it totally surrounds it like a ball. And most of them in cells are going to be rough because we call them rough because they're studded with the ribosomes. And if there are no ribosomes, the ER is called smooth. Both have very distinctive purposes. Rough ER is where we produce proteins because the rough ER holds the ribosomes and the ribosomes make the proteins and the proteins are collected in the rough ER. Smooth ER is totally different. Smooth ER is all about cleaning up, detoxification, if you will. So the rough ER makes all of our proteins. It makes our phospholipids too. And again, it has these little tiny ribosomes embedded on its surface. That's why we say it looks like a rough surface. The smooth ER, same tubules. They extend around and from the rough ER. The smooth ER is very, very busy. Smooth ER is involved with making our steroid-based hormones, cholesterol-based hormones, steroid hormones, the lipids that we use for the lipoproteins, the putting together the triglyceride, the fatty acids and the glycerol to make the triglyceride. We need that. We need those lipids. We make them in smooth ER. We transport fat in smooth ER. we convert glycogen to glucose in smooth ER. Now, two big things that we see here are storing calcium. In our skeletal muscle, we depend on vast amounts of calcium. We depend on vast amounts of calcium to flood our cells, to allow flood our skeletal muscle cells, to allow muscle contractions to take place. Where does that calcium come from? It's stored in the smooth ER that surrounds our skeletal muscle cells. And so we depend on that. We have to have that calcium. Our bodies will raid our bones for that calcium. We make a decision, our bodies have made a decision that it's more important to have our muscles working than to have strong bones. So fortunately, we keep replacing the calcium in our bones too. So that's a very important aspect of the smooth endoplasmic reticulum. When we get into chapter 9 and look at skeletal muscle, you'll see exactly how the smooth ER wraps itself around all of our muscle cells. The other big thing that we see is the detoxification of chemicals. Organs that have vast amounts of smooth endoplasmic reticulum in their cells are the detoxifying organs, like the liver. Now remember that everything we eat and everything that we drink goes into our stomach and then straight into our small intestine. And from there, everything passes through the simple columnar cells into our blood, and from there directly into the liver. And before it can go into circulation anywhere in our bodies, it has to go through the liver and be cleaned up. The smooth endoplasmic reticulum in the liver cells will break down chemicals. It will break down food additives. It will break down pesticide residues. It will break down medications that we take. If we have a backache and we take, a couple of Advils or, ibuprofen, so you take, 400 milligrams of Advil. That's an adult dose. Prescription dose is like 800 milligrams. So let's say that you have chronic back pain or a patient has chronic back pain and they're taking an 800 milligram Advil. Okay. That goes down into the stomach, down into the small intestine, is all absorbed in the small intestine, goes to the liver, and the liver starts breaking it down because it sees it as a foreign chemical. Now, how much of it is broken down in the liver and how much is left to go function in our body? Because ibuprofen, Advil is an anti-inflammatory. It doesn't know where to go. It only works, it hits every cell in the body, and wherever there's inflammation causing pain, it will act on that. So it isn't targeted for any one area. Now, how much of that 800 milligrams gets through the liver and goes to where the pain and inflammation is? It depends. It depends on the efficiency of your liver. It depends on body stature. It depends on a whole lot of things. Well, a typical adult dose, a pharmaceutical prescription dose of Advil would be 800 milligrams. The dose that we get from over the counter is 400 milligrams. So we take the 400 milligrams, we have a headache or our knee hurts or something. So we take that, pain goes away. Or we don't hurt as much as we used to, or our fever goes down. Okay. Working on inflammation. How much of that 400 milligrams got through the liver? Don't know. Enough to make it work for us because we see an improvement in our symptoms. And that dose is based on body size as well as the size of the liver. Now, and that explains why children medicate. A dose for a child is smaller than an adult dose. You wouldn't give 400 milligrams of Advil or 1,000 milligrams of Tylenol to a 6-year-old because A, they have a much smaller body stature, and B, their liver is smaller. And so they could, it couldn't process all the Tylenol or the Advil. And I don't know about Advil, but I know that Tylenol and the liver do not get along well. So you would give a smaller dose to a younger individual because of the smaller stature and smaller liver. The same with an elderly patient. Their dose may be based on the efficiency of their liver and their body size. So this is why how we regulate doses, because it all depends on what gets through the liver. The detoxification process is going to try to break down everything it can. A good example of this is processing alcohol, ethanol. Alcohol can be broken down by the liver at a rate of about 1 ounce an hour. So if you're drinking a beer, you would if you didn't want to have any ill effects whatsoever, you would drink 1 12-ounce bottle every hour. That would be, because that 12-ounce bottle of beer contains 1 ounce of alcohol. If you were having a glass of wine, you would have 1 glass of wine an hour. You would have 1 mixed drink in an hour. And that would allow the liver time to stay caught up with the alcohol. Because when it breaks down the alcohol, it breaks it down ultimately to carbon dioxide and water. Some of the intermediate products that do occur are from the formation of formaldehyde and formic acid and other toxic chemicals. But given an hour to break down 1 ounce, as fast as these toxic chemicals are made, they're broken down. So they occur, but then they're gone. The liver has a problem though with more than 1 ounce an hour. So if your patient ingests 2 ounces of alcohol in an hour, liver can't keep up. They may have some side effects. They may feel a little dizzy. 2 ounces in an hour. Okay, let's just indulge me here. Let's say that they ingest 6 ounces in an hour. Now, in addition to all the effects on the cerebellum and the ability to think and act rationally and walk, correctly, metabolically, the liver is having a hard time taking care of all that alcohol, and it's going to produce these chemicals as it's breaking down, these formic acid and formaldehyde and other chemicals as it's breaking down the alcohol. And it's going to take a while to get caught up. And so a person who drinks, 6 ounces of alcohol quickly, whatever format, or continues to drink, drinks consistently all day or all evening, will often complain of a hangover the next day. The hangover is based on the fact that all these chemicals are gradually being broken down by the liver. People feel, uncomfortable is the best word I can think of the next day because they've had so much alcohol in them that the liver hasn't finished processing. So, because remember, alcohol is a poison. Of course, everything's a poison. A toxicologist will say that everything is a poison, and they're true, because the toxicologist will tell you that the dose makes the poison. Is 1 ounce bad, or is 12 ounces of alcohol bad? Depends. The other thing that happens with the hangover is that alcohol also causes dehydration. As you know what happens when you drink beer, the old joke is you don't buy the beer, you just rent it, because alcohol causes us to pee faster. So the more alcohol a person drinks, the more they have to pee, and that causes dehydration. So on top of dehydration, on top of all these toxic chemicals that the liver is trying to get caught up with, some people that overindulge feel pretty miserable the next day. And there is a risk. A person who drinks excessively in a short period of time can die from alcohol poisoning. It happens. It happens on a fairly frequent basis. It doesn't happen, like every day, but on a fairly frequent basis, we hear about alcohol poisoning, every week, simply because somebody overindulges and drinks, an excessive amount of alcohol in a short period of time. The liver can't keep up and the cells die too fast. Anyway, moving on. So we have the rough ER being brought back into focus now. The rough ER is going to deal with the site of protein production or protein synthesis. Once you make those proteins, you have to collect them. Now, the rough ER will collect a bunch of proteins in a vesicle, but they're all different. At the end of the rough ER, whatever has migrated to the end of the endoplasmic reticulum is going to get collected. It could be hemoglobin, it could be skin color, eye color, hair color, it could be various enzymes. It's all lumped together in a vesicle. That's not going to do us a whole lot of good like that. So we send that vesicle to a structure known as the Golgi Apparatus. The Golgi Apparatus are a series of sacs surrounding the ER that will sort out different proteins and collect them into their own individual vesicles. So a vesicle contains nothing but hemoglobin. A vesicle contains nothing but an enzyme. A vesicle contains nothing but hair color protein or eye color protein or whatever. So they're all going to be organized. The Golgi apparatus, if you think about how FedEx operates and how, like at the Memphis Center, for example, how everything comes into Memphis from all over the East, or at least it used to, from all over the East. And then, planes are unloading with packages from all over going into this. They used to have this huge sorting facility in Memphis. And so everything going to Seattle would go to the Seattle plane. Everything going to Los Angeles would go to the Los Angeles plane, the Chicago plane, the Denver plane. And they're all coming into Memphis. Memphis is the Golgi apparatus sorting out the packages of where they're going to go. And so everything, when a plane's going to Seattle, it's taking stuff that needs to go to the Seattle, or at least to the Northwest. If it's going to Chicago, it's taking stuff that's going to go into the Midwest. So it's taking things where they belong. And that's what the Golgi does. The Golgi looks like this. That's what it looks like for real over here. And this is what it looks like in the drawing. Here comes our transport vesicle from the ER, from the rough ER, containing a mixture of proteins. And as those proteins work their way through here, they are sorted out into their own vesicles, and then they can be released as all hemoglobin at one time, or all hair color or eye color or whatever at one time. And we see that there are 3 ways it can go. We can release the proteins right here, pathway A. Type pathway B, we keep the proteins in the membrane. We don't release them. We don't release them to the outside. We incorporate the proteins into our membranes. Perhaps they are transport proteins. Perhaps they are integral proteins. Perhaps they are peripheral proteins. Perhaps they're enzymes or receptor sites or whatever. Maybe over here they're hormones or hemoglobin or something like that. The third category is that we have produced digestive enzymes, which are still proteins, and we keep them inside their own structure here to dissolve and destroy things that come into the cell, or things that we engulf in the cell, or to take out worn out organelles or worn out structures. But we don't release the contents of the lysosome, that vesicle, to the cell, because it would cause the cell to start falling apart. It would destroy the cell. Now, another type of organelle is the peroxisome. Now this is sort of involves something that we're all familiar with, hydrogen peroxide. You go down to Walgreens or Walmart or Food City or someplace, and you see hydrogen peroxide for sale. It's usually in a brown bottle, usually a rectangular shape or an oval shape. It's usually a brown plastic brown bottle of some type. It says hydrogen peroxide, usually 3%. Well, that's what we use as an antiseptic. We use to clean out cuts. It's good. We use it on the surface of our skin, on injuries. The advantage, of course, is that it doesn't hurt. It foams out. It helps to clean some sort of lesion or trauma or injury to the skin. Well, we use something like that. We produce something like that in our cells. Now, we have to consider substances known as free radicals. A free radical is a molecule that is usually broken apart or somehow or another acquired a charge it's not supposed to have. Consider hydrogen, consider a negatively charged water molecule or a charged glucose molecule. They're not supposed to be charged. And water molecules, they're polar, yes, but they're not. Suppose the water molecule had a negative charge. Suppose glucose had a positive charge or a negative charge. So imagine a cell fragment, a molecular fragment that broke apart and acquired a charge. That's what we call a free radical. They are very reactive. A negatively charged sodium ion would be extremely reactive. We don't want that. We don't want to see that. So the problem is these free radicals will move through cells and destroy tissue. They will damage organelles. They will interrupt normal metabolic activity. And in many cases, free radicals are often associated with development of cancer cells. We talk about antioxidants, like red wine or dark chocolate, things like that. Antioxidants prevent oxidation caused by these free radicals. Oxidation is breaking covalent bonds and releasing energy. Well, that's what these free radicals do, but not where we want them to. So to destroy these free radicals, our peroxisomes use an enzyme known as oxidase. It uses oxygen, and it breaks down all these free radicals into hydrogen peroxide. But it's not the hydrogen peroxide we're used to that we buy at the store. This is very toxic hydrogen peroxide. This is a much higher concentration of hydrogen peroxide than what we get at the store. In the brown bottle, that's only 3%. We're talking something much stronger than that. So, but as fast as we make the hydrogen peroxide, we break down the hydrogen peroxide, the H2O2, with catalase. And that breaks H2O2 down to water. Now, hydrogen peroxide, very simple formula. 2 hydrogens, 2 oxygens, H2O2, hydrogen peroxide. Many scams in healthcare advertise oxygen boosters for their gullible patients, if you will, by giving them hydrogen peroxide injections and hydrogen peroxide tablets. These type of scams tend to run through a pattern where they'll disappear for a number of years and they'll come back again. Because it sounds logical. Hydrogen peroxide has an extra oxygen atom there. It should, in theory, sound good by saying, hey, more oxygen means more energy. Well, it doesn't work that way. In fact, hydrogen peroxide taken as a tablet does nothing. Hydrogen peroxide injected into a vein or intramuscularly may, depending on the concentration, may be may be fatal. So we don't do things like that. Our patients should never be doing stuff like that. So what we use catalase as the second step, we break down the hydrogen peroxide to water. And so it works very well for us. It protects us all the time because, you know, we're usually unaware of all this. You know, the constant battle between the free radicals and the peroxisomes. Also inside our cells, we have an organelle known as the lysosome. The lysosome, I mentioned this earlier, the lysosome are sacs of digestive enzymes held in place by their own phospholipid bilayer in here. These enzymes are going to break down material taken into the cell. You know, if a white cell, for example, engulfs a bacteria, it will fuse the bacterial vesicle with a lysosome, releasing these digestive enzymes on the bacteria and destroy it, or do the same thing to a dead cell, or to a cell fragment, or to a worn out organelle. Lysosomes will take out viruses and bacteria and toxins. It will break down glycogen to release glucose. It will break down bone to release calcium. We do that all the time. We don't like to keep cells around that are non-functional. We don't like to keep cells around that are damaged. And if the cell can't repair itself, it will lyse itself by releasing its own digestive enzymes. And after it starts to break apart, then all of its friends and neighbors will come over and finish eating it. And this is what lysosomes look like. They contain their digestive enzymes inside the vesicle itself, the lysosome itself. Now, problem with lysosomes. There is a genetically inherited disorder known as Tay-Sachs disease. Tay-Sachs disease occurs, it requires a recessive trait from both the mother and the father. It's a homozygous recessive disorder. And what happens in Tay-Sachs disease is an individual receives both recessive genes for this, and that gene prevents the production of a certain lysosomal enzyme that will break down the accumulation of fatty, of a glycolipid, of a fat in the brain. See, during development of the central nervous system, we accumulate certain lipids during this production process, but we don't let them get too excessive. We don't let them get too big. We break them down. But if that enzyme isn't there to break down those lipids, then the lipid accumulates and starts putting pressure on the brain and on the central nervous system, on the spinal cord as it's developing, ultimately causing intellectual, it retards intellectual development. It causes blindness. It causes lack of mobility, and usually kills somewhere between the ages of 5 and 10 in the child. Now they may live to 5 years old. They may live to 10 years old, but usually they die very early because their central nervous system is being destroyed by this accumulation of lipids in the brain and in the spinal column, because this enzyme is missing. So why am I talking about this? Well, it seems that in addition to the genetic component of it, it has a population component. It is predominantly occurs in 3 distinct population groups in the world. Now, originally was found in Eastern European Jews. Historically, Eastern European Jewish populations would live in a closed community, hundreds of thousands of them, tens of thousands, whatever. They would live in an area in Eastern Europe. They would live in cities and towns, and they would stick to themselves. They had a closed population. They would only marry within themselves. Recessive genes like that, because recessive traits are kept alive by you know, they're not inbreeding because it's hundreds of thousands of people marrying and, you know, producing offspring, but it still keeps all of the recessive traits in a smaller subset of the overall population. And so your Eastern European Jews tended to have higher rates of this Tay-Sachs condition. Now, Mediterranean Jews married everybody. So the recessive traits gradually became not non-existent, but it became very rare for it to occur in the offspring of a Mediterranean Jew because of the gene pool was getting larger and larger as more and more people married other people. Not necessarily Jew to Jew, but other Jews are marrying other people. And so the population spread out, gene pool spread out, the recessive traits, the bad recessive traits disappeared. What's that got to do with us? Well, second population subgroup that we see it in are French Canadians, particularly in the areas of Nova Scotia and Newfoundland. They have a higher than normal, higher than background occurrence there too. Again, it's a homozygous recessive condition. So we're getting closer to us. In the 1700s, 1800s, somewhere in that time frame, a large portion of French Canadians migrated to Louisiana and maintained their closed community. The Cajun community, they proudly call themselves Cajuns. The Cajun community can be a fairly closed community. The genetic trait for Tay-Sachs occurs more frequently in Louisiana, in the Cajun community in Louisiana, than it does in any other part of the US, likely, probably, except maybe the Northeast, where a large percentage of Eastern European Jews, their families have, you know, moved there generations ago, but the genes are still there. So, we can see in the Southeast US, we can see and increasing, we can see cases of Tay-Sachs. You know, if you remember from Katrina 20 years ago, we had a large population of individuals from Louisiana migrate into the upper South. So, like in Tennessee, some, you know, here in the Southeast, further away from hurricane country, if you will. So it is a more probable occurrence of seeing this than in other parts of the country, you know, except maybe, like I said, the Northeast. So that's why, you know, I emphasize this, because it is such a unique condition. It is nearly always fatal to children. However, now there are conditions where it can start later in life. It can start in adolescence. It can start in young adults, and it's not fatal there. See, our central nervous system is fully formed somewhere through puberty. Somewhere in that stage and there, our central nervous system is fully formed, and it is no longer affected by the accumulation of these glycolipids. So it's only in our infant stage that we have this problem. So you can be an adult with Tay-Sachs. So anyway, moving on. The cytoskeleton, our cells have structures that we call it the cytoskeleton. It's not like bones and ligaments and tendons like we have, but the cytoskeleton are made up of a series, usually of proteins, microfilaments, intermediate filaments, and microtubules that give our cells their very distinct shape. You know, why are some cells 3 feet long? Why are some shaped like a sac? Why are some skinny and columnar? Why are some cuboidal? It's because of the cytoskeleton structure. What we have here, microtubules, well, spindle fibers are microtubules. Now, they arrange the movement of the cytoplasm and the chromosomes during mitosis. So we see that all the time. We see microfilaments when we're talking about contractile muscle. Actin, the contractile protein that squeezes the cytoplasm apart at the end of mitosis during cytokinesis, is a microfilament. So it's also part of the filament. It's one of the two filaments used, microfilaments used in smooth muscle, in skeletal muscle contraction. And the third type, the intermediate filaments, are the structures like keratin that waterproof our skin. And so we have these three types of arrangements here making up the shape of our cells and doing things with our cells. Also within the cell, is in the cytoplasm, something called the cell center, the centrosome. And the centrosome consists of two structures that look like little barrels called centrioles. And the centrioles are where we generate our spindle fibers from during mitosis, that are going to grab onto chromosomes and pull them apart and push apart the cytoplasm. We have one set of barrels of centrioles in each cell, at least the cells that are going to reproduce anyway. We also see the cell membranes have extensions. We have cilia. Cilia are finger-like projections that move. We use them in our respiratory tract to move mucus up to our throat to swallow it. We use it lining our oviduct in females to move an egg along. When the egg drifts finally into the uterine tube, the fallopian tube is lined with cilia that push the egg along. Flagella are found on sperm cells. Flagella move sperm through semen. Something else we have are villi and microvilli. Villi means fingers, and microvilli means little fingers. These are projections on the surface of cell membranes to increase the surface area. Like the simple columnar cells of the small intestine they absorb all the nutrients. They absorb everything we take in, and they run it all into the liver. I've already talked about that. To increase the surface area, to absorb things, we have folds on the ends of the simple columnar cells. These folds are called villi. The villi have microvilli. These are the villi that you see right here. And then the microvilli are even smaller folds off of that. The total surface area of our small intestine, if you consider it's a tube 20 feet long and about as big around as your index finger, if you were to stretch it out flat to one cell layer thick, it would cover a tennis court. It's a huge amount of space. And that leads us to the nucleus. It's also a great place to stop too, because we're almost out of time. So I will say, okay, just a couple of points. Let me just get to where I went. Let me just go. Yeah, let me go through 3 more slides, and I'll get us to where I am with the other class. The nucleus is the center of the cell, the true center. It contains our DNA. The nucleus has a nuclear envelope, which is its own phospholipid bilayer. It has a structure inside the nucleus called the nucleolus, where it makes RNA. And inside the nucleus itself, in what we call the nucleoplasm, it's the cytoplasm of the nucleus, we have what's known as chromatin. Chromatin is DNA and the protein histone wrapped together. Went the wrong way. So the nucleoplasm is inside the nuclear envelope. The nuclear envelope or nuclear membrane is continuous with the rough endoplasmic reticulum. There are pores, openings in the nuclear envelope that go right into the rough ER. Instructions come out of the nucleus. The DNA sends instructions out in the form of RNA through the pore to the ribosome in the rough ER. And those are the instructions on how to assemble a protein. And the RNA is manufactured inside the nucleoli or nucleolus inside the nucleus itself. It's a dark staining area. And this is what it looks like. You know, there's your nucleus. There's the nuclear membrane or nuclear envelope. Those are all little pores here. And there's a pore opening to the rough ER. There's the nucleolus right there. This is what the pores look like up close. Here they are on the surface of the nuclear membrane right there. And then the membrane itself is made of these filaments that crosshatch across here. The chromatin is 30% DNA, 60% histone, 10% RNA. When the DNA wraps around the histone and it looks like a candy cane, if you will. The nucleosomes are the DNA wrapped around the histones. They're not called chromosomes until the histones, till the whole thing condenses. The chromatin, when it condenses, will form the chromosomes. And they, again, it looks something like this. We see the DNA wrapped around the histone. Again, as I said, it looks a whole lot like a candy cane. But this is where we're going to stop. We'll pick up with this again on Tuesday. And as I think I said earlier, lab this afternoon is the appendicular skeleton. So let me go with a comment here. Lecture test 1. Good question. Lecture test 1 is based on chapter 1, the introduction. Know your body planes, know negative feedback, positive feedback, know the different organ systems that we talked about. The rest of it is going to be on chemistry. Granted, it's only 50 questions, and I tried very hard to make sure that the questions are going to be equally distributed. So don't ignore too much of chapter 1. What kind of imagery is best for bones? X-ray. What kind of imagery is whole body? MRI, that kind of stuff. Yeah, my first few lectures. Exactly. The second part of the test is chapter 2, all about the chemistry. So it will be multiple choice, but it won't be 26 choices like that. No, I'm not going to be, it won't be like the lab test. I will cut it down, so don't worry about that. So study chapter 1 and study chapter 2. Make sure you know what a lipid is, it's a lipid. A fat is a glycerol and 3 fatty acids. Know what an unsaturated fat is. Know what a saturated fat is. Know what a carbohydrate is. Know what a monosaccharide is, a simple sugar like glucose. A disaccharide is 2 sugars like sucrose. And a polysaccharide is glycogen in humans and starch in plants. The proteins, we talked about proteins and denaturation. Proteins are amino acids linked together. And then we talked about the DNA and RNA. Make sure that you know how many electrons are in the outer shell. Because I know that there are questions on there about, if you have X number of electrons, how many electrons are in the outer shell of, this particular atom? Or if you have X number of protons, how many electrons are in the outer shell? We know that protons and electrons are the same number. So things like that. Make sure you remember that. You can process that. So, any other questions? If you have any questions, email me. I'll be glad to answer them for you. Let me get out of this. Stop sharing. Don't worry about that. We didn't talk about palpations and sounds. That's more of an AP2 type thing anyway. So that we're not going to worry about listening to the heart sounds. We've got enough to learn to remember for the test. Any other questions? Okay, then I'm going to get us out of here. I'll see some of you, I guess, in lab today, and the rest of you see next week. Have a great weekend.